Clinical chemistry, lipids and lipoproteins. Lipids or fats are hydrophobic. Hydro is talking about water, phobic is talking about the fear of water or they repel water. Nonpolar organic molecules. They're complex alcohols that combine with fatty acids to form esters. They yield fatty acids on hydrolysis. So hydrolysis means to break apart using water. So when they are broken down, they're broken down into fatty acids. They have a diverse structure and function. Some serve as membrane components, bile acids, steroid hormones, surfactants, vitamins such as A, K, D, and E, and they're a rich source of energy. Clinically relevant lipids are listed below. You can take a look at the sterol derivatives, fatty acids, the glycerol esters. Probably the most common glycerol ester that you would have heard of is a triglyceride, sphingosine derivatives, and so forth. If we look at the concentration in plasma, triacylglycerides or triglycerides have the highest concentration, followed by cholesterol, phospholipids, and then fatty acids. Triglycerides contain 3 fatty acid molecules attached to 1 molecule of glycerol. 95% of tissue storage fat is triglycerides. The primary function is to provide energy to the cell. They are uncharged or neutral lipids, and the predominant glycerol ester in the plasma. They're transported in lipoprotein molecules. Cholesterol is an unsaturated steroid alcohol containing 4 rings and a single side chain tail. It's synthesized almost exclusively by animals and not readily catabolized by most cells. It is not a source of fuel. Phospholipids are similar to triglycerides except only 2 esterified fatty acids. Those are known as the hydrophobic tails. The third position on glycerol backbone contains a phospholipid head group, and the head groups contain choline, inositol, serine, ethanolamine, and all of which are hydrophilic in nature. Hydrophilic, water-loving. Just to kind of backtrack on the purpose, so we talked about the purpose of triglycerides is to provide energy to the cell. The primary function of cholesterol is to comprise the cell membrane, so that provides rigidity to the cell membrane. Also, cholesterol serves as the basis for steroid hormones and other types of things that we use in the body that are steroid-based. Phospholipids are going to make up the cell membrane. And then fatty acids. So fatty acids are linear chains of carbon-hydrogen bonds terminating in a carboxyl group. What's pictured here is an unsaturated fatty acid. Saturated fatty acid. Saturated means that it contains all of the hydrogen bonds that it could possibly hold. Unsaturated means that there is a double bond somewhere in the chain so that it does not contain all of the hydrogens that it could possibly hold. So those double bonds cause hydrogens to be lost. So anytime that you have a double bond, it causes a kink in the chain, and that's why typically our unsaturated fats cannot be solid at room temperature. They're more so like the plant oils and such. There are some essential fatty acids. Essential fatty acids must be supplied in the diet. So someone that is trying to have a zero-fat diet is going to be missing out on these essential fatty acids. Linoleic acid, linolenic acid, and arachidonic acid. Fatty acid transport. So free fatty acids are typically bound to serum albumin. 1 albumin can bind up to 20 molecules of fatty acid. As esters with cholesterol or glycerol, most fatty acids are going to be in combination with glycerol. So we have monoglycerides, diglycerides, and triglycerides. Lipoproteins. Lipoproteins are complexes of lipids and proteins. They're designed to transport lipids and facilitate metabolism in hydrophilic environment. So if you think about the bloodstream and how much water is inside the bloodstream, now imagine putting fats directly into the bloodstream. If you've ever seen a bottle of salad dressing, you know that if there's oil in there and water in there, those layers are going to want to separate away. The fats are going to want to combine together, separate from the water, because the fats are hydrophobic, the water is hydrophilic. When that happens, and if that happens in the bloodstream, that can create a whole bunch of issues. So in order to transport fats through the blood, we have to complex these fats with proteins, and that allows them to be transported. So lipoproteins are just that. They're proteins combined with lipids that allow for the transport of lipids through the blood. Apolipoprotein, or APO, is the protein component, and that's free from the lipid. There are different types of apolipoproteins, or APOs, and those will be discussed on the next slide. Lipids are bound loosely enough to allow for ready exchange of lipid between serum tissue lipoproteins, but strong enough to allow for physical separation by analytical systems. So here you can see in a typical lipoprotein, there's phospholipids, the cholesterol, some triglycerides, and there's the apolipoprotein. So this is a list of the apolipoproteins. So there's different types that exist. So A1, A2, B48, B100, C1, C2, C3, and E. And each one of these is responsible for transporting different types of lipoprotein classes. So A1, HDL and chylomicrons, A2, HDL, and so forth. So you can review this table, take a look at it, and then review the functions of each of these as well. So the 5 classes of lipoproteins are chylomicrons, VLDL, or very low density lipoprotein, IDL, intermediate density lipoprotein, LDL, low density lipoprotein, and HDL, high density lipoprotein. You can see the densities that exist. The main components, so chylomicrons, are primarily made up of triglycerides. VLDL is triglycerides, IDL is triglycerides and cholesterol, LDL is cholesterol, and HDL is protein. You can see the apoproteins that are involved and the relative diameter. So just to kind of review where cholesterol comes from and where fats come from in the diet. So when we consume fats, we're going to break it apart, break those bonds. We have an enzyme called lipase. Lipase is released by the salivary glands, but then again by the pancreas. We have salivary lipase and pancreatic lipase. So once the pancreatic lipase combines, we're also going to have bile present, and bile comes from the gallbladder, which was produced in the liver, stored in the gallbladder, released into the duodenum of the intestine. So once it gets into the intestine, you're going to have fats being digested. So lipase is actually going to break the bonds, and then the bile salts are going to surround those broken apart fat molecules. And we're going to break it down into fatty acids and glycerol, so the bile salts are going to separate those and surround and separate those molecules from one another. When the bile salts do surround those components, it's called a micelle. So here you can see the fatty acids are being separated from the glycerol, and the bile salts are going to be surrounding those components. Those components in the micelle are then brought to the intestinal wall, where they're going to be absorbed by the intestinal cells that surround that intestinal wall. So the individual components are going to be absorbed, and then they're going to reform into triglycerides, where they're going to be packaged into chylomicrons, and chylomicrons are going to transport the triglycerides through this exogenous pathway, so from the outside of the body to the inside of the body. Exo means outside. Exogenous pathway, these chylomicrons are going to bring them internally, and for the most part, they're going to go into a lacteal. The lacteal is part of the lymphatic system, and then they will be routed to the liver from there. So here's kind of the rest of the pathway. So from the intestines, you have that exogenous pathway that I mentioned, where chylomicrons are going to bring the triglycerides to the liver. From there, they can be stored in the liver, they can be converted to different substances that are needed in the body, or they can be transported to the tissues. So if they want to be transported to the tissues, VLDLs are going to transport those triglycerides to the tissues, and that's known as the endogenous pathway. So VLDLs can transport triglycerides to the tissues, and then we have LDLs and HDLs. LDLs are going to transport cholesterol to the tissues, and HDLs are going to transport cholesterol back to the liver to be degraded. So if you start thinking about like the good cholesterol or bad cholesterol, cholesterol is just cholesterol. We have good cholesterol transporters and bad cholesterol transporters. So LDL, low density lipoprotein, is one of the bad cholesterol transporters because it's carrying cholesterol to the tissues to be deposited in the tissues. And that's a bad thing. We don't want a lot of cholesterol being deposited in the tissues. Low levels are needed for cell structure and integrity and for the creation of certain steroid hormones. But once we start getting too much cholesterol transported to the tissues, we can start to have it accumulate inside of our arteries and in places that we don't want the cholesterol to accumulate. And we will talk about that and the progression of heart disease a little bit later as we go through these lectures. So LDL is associated with being bad because it's going to be putting that cholesterol into the tissues. HDL is good. Now remember, cholesterol is just cholesterol. It's not like there's good types of cholesterol and bad types of cholesterol. It's just cholesterol. The transporters are what are good or bad. HDL is considered good because it's going to transport cholesterol back to the liver to be broken down. The more HDL you have, the more protective it is against things like coronary artery disease or atherosclerosis. So now just going through each one of these and the details. So we talked about the pathways that these would travel through. So now we're going to talk about the details specifically. So chylomicrons, if you remember from the diagram, and I'll go back to the diagram, chylomicrons, once the triglycerides are absorbed from the diet, chylomicrons are going to transport those triglycerides to the liver. So the lipid component, you can see the breakdown. So predominantly triglycerides. The protein component is 2% by weight. Source is the intestine. They're generally not present under fasting conditions. Presence makes serum appear milky or turbid. Lipemic is what we would call the serum if it looks milky. It will form a creamy layer on the top of the serum upon standing. They function primarily in the transport of exogenous triglycerides. So you should know what exogenous means. So exo means from outside. Genus is where it arises from. So exogenous triglycerides would come from the outside of the body. So we're talking about the digestive tract. These are going to be absorbed from the diet. Very low density lipoprotein. If you remember what VLDL does, going back to this diagram here, our chylomicrons took the triglycerides to the liver where they're going to be processed and put where they need to be. If the triglycerides are in excess and they need to be stored in the tissues, VLDLs are going to transport triglycerides through that endogenous pathway. So VLDLs are cholesterol, triglycerides, and phospholipids. The protein component is 8%. The source is the liver. Primary function is the transport of endogenous triglycerides. Endo means from inside. So these are triglycerides that were from inside of the body. Intermediate density lipoprotein, or IDL. The lipid component is 85% and the protein component is 15%. So you can see cholesterol, triglycerides, phospholipid breakdown here. It is the precursor of LDL, and its source is from the catabolism of VLDL. It's a transient particle that's usually present in low concentrations in fasting plasma. Low density lipoprotein. So the lipid component is 79%, the protein component is 23%, and you can see it has a lot of cholesterol in it. So it constitutes 40 to 50% of lipoprotein mass in humans. The source, it's made from the catabolism of IDL. Its primary function is cholesterol transport. It's atherogenic. Small or more dense particles are more atherogenic. Oxidative modification results in atherogenicity. Atherogenic is talking about it's going to be able to produce some effect on the artery walls. So atherogenic, it's going to, in other words, start packaging the cholesterol into the artery walls. High density lipoprotein, lipid component is 50% and the protein component is 50%. The source is the liver, further fractionated by ultracentrifugation into HDL2 and HDL3 fractions. HDL2 is present in premenopausal women at 3 times that seen in men. Lower levels are associated with increased premature congestive heart disease, and higher levels are protective against congestive heart disease. Four major pathways involved in lipoprotein metabolism. So lipid absorption. In the digestion or digestive tract, dietary lipids are converted to amphipathic lipids. Amphipathic lipids form micelles in the intestinal lumen. Micelles come in contact with microvillus membranes of the intestinal mucosal cells and absorbed. Then we have the exogenous pathways. So we already talked about these pathways, but these are just providing additional definitions. So chylomicrons are going to interact with the proteoglycans on the surface of capillaries in various tissues in circulation. Free fatty acids and glycerol from hydrolysis of triglycerides by lipoprotein lipase can then be taken up. Then we have the endogenous pathway. VLDL loses core lipids, causing dissociation and transfer of apolipoproteins and phospholipids to other lipoprotein particles. During this, VLDL is converted to VLDL remnants, which can be further transformed by lipolysis into LDL. Half of VLDL is converted to LDL, and the remainder is taken up as VLDL remnants by the liver remnant receptors. Then we have the reverse cholesterol transport pathway, where HDL removes excess cholesterol from the cells.